years ago, I was at a conference. It actually wasn't even a neuroscience conference, but I was discussing my children with someone and how their situation has led to my mission in neuron regeneration. And they were like, "Oh, like the nerves in our nose." And I was like, what do you mean? What are you talking about? And they're like, "Yeah, I heard somewhere that the neurons in our nose, they heal or regenerate or something like that, different than the rest of our body." And so I looked into it, and sure enough, olfactory neurons are highly regenerative throughout adulthood. And so, that got me really excited. And very recently, there was a published review paper titled Olfactory Receptors and Neural Regeneration in the Central Nervous System. And that really re-sparked my interest in this topic. And I knew I just had to get one of those authors on this show. And in fact, I actually got two, who you'll meet in just a moment. But before we dive into that, I just want to give you a little idea of what's to come, or, what you find in this paper by reading a few sentences from the abstract here. The ability of the olfactory system to regenerate its sensory neurons provides a unique model to study neural regeneration, a phenomenon largely absent in the central nervous system. Insights gained from how olfactory neurons continuously replace themselves and reestablish functional connections can provide strategies to promote similar regenerative processes in the central nervous system, where damage often results in permanent deficits. So, my guests today are two of the paper's authors, Claudia Garrigós, who's a PhD student, and Jaume Lillo, who's a postdoc, and they're both at Dr. Rafael Franco's lab at University of Barcelona. They both provide their backgrounds in the beginning of the interview, so for the sake of time, I won't read their full bios, but I do strongly suggest you check those out at our show notes for this, where we'll also link to the full-length text of the review paper that we're digging into, and you can check all that out at neuronregeneration.com/10. Before we get into the recorded interview I did with them, I do want to mention that Claudia is looking for a U.S. lab to do a 3-month exchange during her PhD program, for which she already has funding. So if anyone is listening to this and is interested, if you go to those show notes, you'll also be able to click through and find her LinkedIn profile and connect with her there. Again, that's neuronregeneration.com/10, the number 10. So with all that said, let's dive into the exciting world of central nervous system neurons that readily regenerate throughout life. Welcome, Claudia and Jaume. Thank you so much for taking the time. I am so excited to finally get into this conversation about olfactory neurons. Before we do, why don't you both give us a quick background on yourself, your work, and the lab that you're working in right now?
Okay, so I'm from Alicante. It's a smaller city in Spain, and I studied biology in the degree. And afterwards, I came to Barcelona to study biomedicine. And I've always had interest in neurodegenerative diseases, mainly Alzheimer's disease, but also in others such as Parkinson's disease, etc. And right now I'm in the molecular neurobiology laboratory here at the University of Barcelona in the Department of Biochemistry and Molecular Biology in the Faculty of Biology, and in the group of Dr. Rafael Franco, which is focused on GPCR receptor, and they're studying modulating neurodegenerative diseases mainly. So that's my primary interest right now. I'm doing my PhD, so it's the beginning of my career, and it's exciting.
That's very exciting. Yes. And thank you again for taking the time. I know the life of a PhD student is certainly very busy. Can you, for those who, we have a broad audience and plenty of non-scientists out there, can you give us the name of the type of receptor, like spelled out, that you just mentioned that y'all are looking at?
Well, I'm mainly focused in the olfactory receptor 51E2.
Okay, great.
R51E2 is
Okay.
what I'm working
And
with.
those are types of you said the GP. I'm mixing up the
GPCR, G protein coupled receptor.
That's right,
Yeah.
G protein coupled receptor. That's what I was going for. Okay, perfect. Okay, Jaume, how about you?
Hi, thank you for inviting us.
Sure.
Well, I'm Jaume Lillo, and I'm currently doing my postdoctoral stage here in Barcelona in Rafael Franco's lab in the same group with Claudia. And my research is the same. I mean, it was focused on GPCRs, G protein coupled receptors, and its implication in neurodegenerative diseases, We worked in the laboratory with adenosine receptors, dopamine receptors, also cannabinoid receptors. And I also studied the neuroprotective effect of some cannabinoids, such vidiol and cannabigerol, that nowadays are very famous right now.
Yeah.
And these last years, we focused also in the olfactory receptors that are also a type of GPCRs. And we studied this, we found interesting these receptors because they are found in different types of cells in the central nervous system, such as neurons and glial cells, not only in the olfactory epithelium. And we were wondering why those receptors are in the brain if they are not smelling when they are not found on the olfactory epithelium. So we try to focus on this topic, since they are also neurodegenerative diseases that have affection on the sense of smell, for example, in Alzheimer and Parkinson's. And now we are currently with Rafael Franco investigating this topic.
Yeah, and I found it so interesting that those types of olfactory receptors, right, are literally found throughout the entire body, right? And they have
Yes.
and they function throughout the body. They're not necessarily smelling things, but what a cool discovery! And it seems like, in looking at the review paper that we're kind of digging into today, there could be really big implications for the fact that they're fairly ubiquitous throughout the body. So could one of you give just kind of a general, we'll get into the details, but a general summary of that review paper? And I'll go ahead and just read the title of it for those listening in: Olfactory Receptors in Neural Regeneration in the Central Nervous System.
Yes. So a small summary about the paper that you just mentioned. The paper is a review, and we wanted to do a review because, as you said, it's a very interesting topic, and it's cool that we can put all this information into one place so more scientists can go ahead and find information about it. And it mainly dives into, even if the receptors are important, olfactory receptors are important for smell, they help us detect odor and molecule. But even if that's interesting throughout evolution, we can also find them, as Jaume mentioned, ectopically, which means in unexpected places or performing some role that they are not described mainly for. And our review explore how these ectopically expressed olfactory receptors might play a role in helping the central nervous system regenerate after injury or in the context of neurodegenerative diseases. And we explore the idea that the olfactory system has a unique ability to regenerate neurons and throughout life, and that might hold clues for promoting similar regeneration in other parts of the central nervous system where the damage is sometimes permanent.
Yeah, and so from what I've learned before this and from your paper, there's really only kind of three primary areas where we find adult neurons will actually regenerate and heal and replace themselves when they're damaged, and olfactory being one of them, right? So I kind of like want to get straight to the big question, which is, do we know why and how neurons of the mammalian olfactory system, that they can regenerate throughout life when most neurons in the CNS can't? What makes them different, and do we know how they do this?
Well, that's the million-dollar question.
Yeah.
And the olfactory system is the one that is more exposed to damage. The neurons in the olfactory epitheliums are closer to other environmental factors that can affect them, thus making that throughout evolution, we probably have found the need of making them regenerate, which is very interesting. But also there are a few key factors that help this process happen, such as the presence of stem cells. The olfactory epithelium has stem cells that can differentiate into new olfactory sensory neurons. Then also, as we mentioned in our review, we have the glial support that are the olfactory and sheathing cells. That is a special type of glial cell that provides the support that these neurons need for performing that specific process. And also that is molecular mechanism that is unique, and it involves things like cell adhesion and signaling pathways that facilitate the regeneration, but it's still a big question mark in trying to like solve all the details of this process. That's what makes it more interesting.
Yeah. So, so you just to kind of summarize, make sure I understand, there's this presence of olfactory and sheathing cells, which are essentially stem cells, right? And then also olfactory and sheathing glial cells, the support cells. And then you said that the third part was that there are some pathways that are unique in that area that are a part of this regenerative process. Do we know, am I saying that right? Or do we know like specifically what pathways are there that are unique to this regenerative process compared to other parts of the CNS when there's damage?
No, I mean that more specifically that the regeneration of the neurons is what is unique in there.
That's okay. That's the unique part. Yeah.
That meant, yeah.
And so is it known, and maybe it's not, and you're saying like that's kind of the $1 million question, but other than those types of cells being present, the stem cells and then these specific types of glial cells, is there anything known about the pathways or just the process after there's damage that's unique that we might then be able to try or maybe people are already trying to say, well, can we apply that, say, in the spinal cord after SCI, or can we apply that out in the brain after a traumatic brain injury?
Well, I think the main idea of this regeneration of the olfactory epithelium, thanks to the olfactory sheathing cells, right, that Claudia mentioned, that those are the cells that are mainly used in the treatment of spinal cord injury because they have specific functions. For example, you have to imagine those cells. They are different kind of glial cells. In the central nervous system, we have astrocytes, oligodendrocytes, microglia, but these glial cells are specific of the olfactory epithelium. And you have to imagine those cells like a bridge that is connecting the olfactory epithelium and the olfactory bulb. And it's guiding the axonal growth of these stem cells that are going to become a new neuron in case of damage. So this property of these olfactory sheathing cells, that it's the support and the guidance and allows the direction of the correct direction of the axonal growth, it can be used in a disease like a spinal cord injury, because you can use these cells in order to try to guide also the axonal growth and try to reconnect these neurons that are damaged. So that's the main idea of the use of these olfactory sheathing cells in this kind of therapy.
Okay. And yeah, I mean, that actually leads to another question that I had is in your review paper, you had a section that kind of describes that there's preclinical research. So in cell cultures and animals, I assume, using these olfactory and sheathing glia and also OECs, the stem cells, with positive results, I think, for I saw spinal cord injury, multiple sclerosis, and Parkinson's. Can you maybe elaborate on some of that work in terms of what results that they got in those different types of syndromes? And I know that this is preclinical animal or cell models, but maybe just let the listeners know kind of what they've been able to achieve in those models using these olfactory cells and sheathing cells and glia.
Yeah, well, one of the a part of this thing that I told you, one of the good properties of the olfactory sheathing cells is that they promote neuronal growth, okay, and they also have remyelination capabilities. I mean, they can enhance the myelination, the myelin, that is something that is lack, for example, in multiple sclerosis. So you can also use these olfactory sheathing cells also in models of multiple sclerosis and try to remyelinate the neurons that are damaged. So the main studies that were performed, it's basically in multiple sclerosis and also in spinal cord injuries, and it was performed in mice where they try to implant, do a transplant of this kind of glial cells, and they were good outcome in those experiments. But I mean, there's a difficult field and there's more to be done yet. But yeah, they were quite promising the results in those.
And with the SCI work, was that done fairly recently, and there's a lot of ongoing efforts to maybe translate it outside of animal models, or was that a long time ago? What kind of is the status of that work?
I believe that there's still not a big clinical trial in humans, as far as I know, but it was very good because the results showed things that even if the damage was already done, the glial cells were able to like recover that damage, which is also important because it's not the same as having the cells and then causing the damage to the, I think the area was the hippocampus, but I will have to double check in order to like say it 100%. But in after doing the transplant, as Jaume mentioned, they saw the recovery, which is what makes it like pretty interesting. But olfactory neurons and olfactory receptors are a very difficult field because you have difficulties, for example, finding, in the case of olfactory receptors, that are the, like after, as Jaume mentioned, the olfactory neurons are activated by the odorant molecule, and then the signal passes through the nasal cavity and it goes to the olfactory bulb that is in the skull. Then the olfactory receptors are activated, and then they do a series of signaling pathways, such as increasing cyclic AMP levels, for example. And all this is very difficult to study because outside of the olfactory epithelium, there are many molecules that can activate these odorant receptors. So that makes it very difficult to study. So that's why you cannot find, is a topic that is very interesting, but is not as maybe other type of receptors that are more known and more well known, because you need a lot of time in order to like make progress in this field,
Mhm.
because they are very difficult to like overpress when you have like heterologous systems. They are difficult to spread. So it's a difficult field. That's why the results are not like, every now and then,
Yeah,
but
yeah. There's
more
not a
in
lot of
time.
people working on it. Yeah.
Hmm.
It seems like, well, just on the topic of spinal cord injury in general, a lot of a lot of difficulties, getting things to translate out of preclinical into humans. Just a long history of difficulty taking exciting results from the lab into humans for sure. But so there's not a whole lot of people focused on it because it is quite difficult, but there is ongoing work in the field in SCI.
Yeah.
I think I saw Parkinson's as well. That's exciting with the multiple sclerosis and remyelination results. Do you know if is anyone trying or looking at or anywhere close to maybe taking that into human trials for MS? Do you all know?
I'm not sure, but I read a lot about the in human. I searched for human clinical trials, but I just found it in spinal cord injuries. I'm not sure if there's any in terms of multiple sclerosis, but probably yes. But I mean, maybe now it's more focused on these spinal cord kind of injuries.
Mhm. Yeah. Okay, cool. So, what do you guys feel like are the biggest challenges, since just kind of to continue on that thought process of translation into trials and into humans? What do y'all feel are the biggest challenges when it comes to translating this particular science with olfactory neurons and their regenerative capabilities into humans? And Claudia, I know that you just kind of mentioned there's a lot of different things that activate them, throughout the body. And so it's hard to kind of study. So that would be one big challenge, obviously. Is there anything else that you feel like is kind of holding researchers back or creating a roadblock to getting to a point where we could take it into humans in different syndromes?
Well, I think that the main problem is also the problems that you can find in different kind of transplants. So for example, if you are talking about the spinal cord injuries, okay, but you also have to take in consideration that you have to study if it's the safety of this process in the long-term impact of this transplantation, okay? Because there are plenty of studies when you are transplanting any other organ, but maybe there are not a lot of studies in terms of the long impact of this olfactory initiating glia, for example, the kind of transplant, okay? You also have to obtain a large number and high quality neurons in order to do this transplantation, and you have to clinically deliver these neurons. So the technicity is quite challenging.
Yep.
And I think maybe also it's difficult to measure the functional improvement in this kind of pathologies. I mean, because scientists quite new this field, you have to create new tests, or you have to find a way in order to be sure that the process is working, right? And you have to take in consideration that there is, like in other kind of transplant, there is a huge variability between individuals in terms of age, health, the injury severity. Not everybody has the same spinal cord injury, for example. And also the most important part, the immune system. Not everybody have the same immune response. So I think that's something that has to take into consideration. It's quite difficult in order to study.
Yeah, yeah. Well, there's a lot of roadblocks, a lot of stuff. So as you guys were doing all this, obviously extensive research and reading to put this review paper together, I'd love to hear what papers or science that you went through. I mean, the bibliography is quite long, you know. And so what of those did you find personally most interesting, fascinating, exciting as you were doing this work? And I'd love to hear from both of you on that one. Yeah.
For me, the fact that the work saying that the olfactory receptors are expressed not just in neurons but also in glial cells like microglia and astrocytes, it was really like eye-opener. Like, it was very interesting for me because also as I mentioned, the olfactory receptor that I work with, I work in a context of like neurodegeneration, as we mentioned, and like inflammation and more in like a molecular level. But this receptor is known for being a marker in prostate cancer. So that makes it more interesting how a receptor that is expressed in a different tissue and with different quantities and regulations can be also useful in a different context of pathology.
Wow, I didn't come across in the paper or know that it's actually a marker in prostate cancer. Great. Jaume, what about you?
Yeah, well, when we were we are quite new in the field of olfactory receptors, so this review was also a practice for us in order to learn and read different things. And there's a part in the review that talks about the evolution of the olfactory receptors, and I was quite surprised of that these olfactory receptors appeared more than 700 million years ago, and it was quite fun to follow the evolution of these receptors, and in fact, knowing that one of the oldest animals with orthologs of these olfactory receptors, it was a medusa. So it was quite fun to know this. And yeah, I really enjoyed it, reading all these parts of the evolution of the receptors and olfactory receptors. And quite interesting. Yeah.
So you're saying that because and maybe I missed that part. So they were able to trace back, and they feel that the first appearance of the olfactory receptor was in a medusa, like a.
Well, I'm not sure if it's the first. I can't really tell, but yeah, it was evidence of an ortholog of a kind, a more simple kind of olfactory receptor. Okay, but it was also present in medusa, because if you think about it, the sense of smell, it's necessary for evolution, right? You are constantly smelling, so it makes sense that this kind of receptors appeared a long way ago, and it has been between us for many millions of years ago. Yeah.
Wow, that is fascinating. Great.
Yeah.
So maybe I'll finish up with this one. Who are your scientific heroes? I'd love to also hear from both of you on this.
Well, since now we're talking about evolution, no, yeah, but I think there are many candidates to be in the top one, but I think that my hero it should be Darwin, because I think he was able to achieve so much with so little. Okay, that imagine just by observing animals and the work, the implications of his work that are nowadays are so important. It was not only important in ecology, I mean in the evolution of species, but also in the evolution of proteins or genes. And I think his work was amazing, and I really admire Charles
Great,
Darwin.
great. Claudia.
I agree with Jaume, but I will also like to add Ramon y Cajal because, well, I'm also a big fan of painting. I paint in my free time other than a scientist. So, and I love his work and the neurons and the paintings and how detailed studies were on neuronal structures and how much that has impact modern neuroscience and demonstrating the organization of the nervous system. That was
Cool.
very cool.
What was the name? I want to look into him. What was the name again?
Ramon
Ramon? Yeah.
Cajal
Lahal? How do you?
is, I can spell it,
Yeah.
is Ramon is Ramon
Yeah.
Ycajal.
Okay. Cool. I'll check him out. Very good. All right. Well, thank you both again so much. Fascinating stuff. And though obviously there are, a lot of hurdles to overcome and things to work through in trying to translate this into humans, it's just so exciting to know that there's this type of adult neuron that is so regenerative. And somewhere in my gut, I just feel like there's something there that potentially we someday will be able to apply to other parts of the nervous system and kind of hijack that regenerative capability. So yeah, thank you for the work you were doing. This is a great review paper. We'll definitely link to it in the show notes. So hopefully more people will be able to utilize it and continue speed their work through the work that you guys have done. So thank you.
Thank you so much.
Thank you for inviting us and for giving us this opportunity of sharing science with more people, which is a very great work in order to like get more people to know what we do and how important that is for the world. So thank
Absolutely.
you.
Yeah, no problem. My pleasure.